Hi, I'm Mrs. Murdock, and today I'm going to talk about experimental design. Experimental design. Remember, we talked about the scientific method, and this is the point in the scientific method where you have asked a question, formulated a question, and formulated a hypothesis, and now you need some way of testing if your hypothesis is supported or not. So experimental design is really the bread and butter of being a scientist. If you know the intricacies of experimental design, if you understand how to design an experiment, then you're much better equipped to understand other people's experiments. You're much better equipped to understand what somebody's talking about if they did a study of something and they have some data. And then you're more prepared to ask questions, and that's very good for you because you have a better understanding of that. It's also very, very much a part of science class, and you'll do better in your biology or any kind of science class if you have a really good understanding of this. So the type of experiment I want to talk about is an experiment that would be controlled in a lab. So a controlled lab experiment where you are working on test subjects in a laboratory, in a controlled environment, is very different from other types of studies that are also done in the field, where you might be studying a living organism out in its natural habitat. And there are arguments for both of those types of experiments. The argument for a field study, where, if you want to look at a bunch of mice and what they're eating in the wild, you'd be best to do that if you go out and actually observe them doing what they're doing in the wild. But then you have much less control over other variables that might be affecting their diet. On the other hand, if you have a controlled lab experiment, you can bring those mice into the lab and you can give them food choices, and you can absolutely control all the other variables, and you can isolate just what you want to test with them. But there's pros and cons for both. In any case, today, the controlled lab experiment. So let's pretend for a moment that you are working for Green Grass Incorporated, and they are all about making fertilizers for people who want to grow plants, or especially for people who are interested in having nice green thick lawns. That's a big thing here in the suburbs where a lot of us live. So you have just been tasked with testing a new chemical that they think is very good at increasing the rate of grass growth. And you are given a big lab, and you're given this big assignment where you're supposed to go in and design an experiment to test the question or test the hypothesis. You're starting with a hypothesis. All labs start with that. And your hypothesis is we'll call it chemical X, does chemical X increase the growth rate of grass? Now, you can be much more specific than that. We would probably, if you were really doing this and you were really working in a corporate or university situation, you would be specifying the chemical. You would be specifying a lot of other things with this hypothesis, the species of the grass and such. But we're just going to keep it simple today. So you've got this hypothesis, and the job of an experiment is to test the hypothesis. So the first thing that you want to do is you would want to get some test subjects, right? So you're going to need test subjects, and your test subjects are going to be grass plants, right? Grass plants or grass shoots, young grass plants. Okay, and those test subjects, you're going to need to subject them to different conditions. So you may remember the terms independent variable and dependent variable. In this case, your independent variable which is what you're going to subject your test subjects to. Your grass shoots, you want to treat them with different levels of fertilizer. So your independent variable that you are manipulating is the amount of fertilizer, or amount of chemical X, right, that they want you to test. Amount of chemical X that you're giving to your test subjects, your grass plants. And then your dependent variable, which then would depend on your independent variable, would be their rate of growth. Their rate of growth. So then you would want to set your experiment up. So you would need, let's assume that you have a lab that's a nice big lab and you have the possibility of doing lots of things here, but you would need, some pots. And let's just set up a simple example here. I'm trying to make all the pots the same size. I'm doing the best I can with that, because that's an important thing that we'll talk about, is that everything should be standardized when you're testing one thing. All right, and in the pots you're going to have soil, and each pot's going to start out with grass shoots of approximately the same age that were germinated and started to grow at the same time, right? And you're going to want to have different amounts of chemical X in each one. So you're going to want to have one that has no milliliters in one. And then maybe one that has 1 milliliter, and then maybe one that has 5, and then maybe one that has 10 milliliters of your chemical X. Okay. So your 0 milliliter, you may already be on top of this and thinking in your mind, that's the control. Yes, that is your control subject. And these, all three of these, there happen to be 3 levels of test subjects. Right? Okay. And so you set your experiment up like this. Well, the reason why you have a control subject, and you may already remember this from your 7th grade science, or maybe even earlier, that you want to make sure that you have some test subjects that aren't receiving your treatment, because that gives you a way of comparing. I want to see what the effect of this chemical X is on the growth of the shoots, but I also want to make sure I have an example shoot that I didn't do anything to. It didn't get any of the chemical X. So if there is, a whole bunch of growth with these 3 test subjects, the 1 and the 5 and the 10 milliliter, but if I didn't have a control test subject at all, I just had these and they all grew a lot, how would I know that that was more than if I hadn't used any at all? So the control subject is a way of making sure that there is an effect, that your treatments, your chemical X treatments, are actually having an effect that's different from if you didn't use a treatment at all. That's the value of having a control. The other very, very important part of a controlled lab experiment, and the advantage of a lab experiment, is that you have something called constants. You can keep all other variables around these plants constant. Everything else that you're doing with these plants, except for the amount of fertilizer you're giving them, is the same. So you keep all other environmental variables the same. So what kind of variables would those be? In this case, what would you keep the same for all of these? You would keep the species of grass. Would have to be the same. The type of soil. The amount of water. The amount of light. The temperature around the plants. You would want to keep everything. And if you're in a lab, if you're in a room that's nice and big and you have a lot of space, then you have the capacity to do this. And so that gives you a lot of control. So then let's say that you collect some data. And let's let's say you collect some data here. And the data kind of looks like, let's say that the data doesn't look, the data looks awesome. Let's say the data looks just like this. Let's say that after a while you get a bunch of data, and I'm going to make a graph here. And on the x-axis you have your amount of chemical X, and over on the y you have rate of growth. And let's say that you have, here was 0, and here was 1, and here was 5, and here was 10 milliliters. Forgive me for not adding units. And let's say that your 0, your control plants grew this much. And let's say your 1 milliliter plants grew this much, your 5 grew this much, and your 10 grew this much. So it definitely looks like there is an effect of your chemical X on the growth, because you definitely have more faster growth in the plants that got fertilizer than the one that did not. So you go to your boss and you show her the data, and she's like, well, so how many plants did you actually test in your experiment? And you say, oh, well, I had 5 plants within each group. And she kind of looks at you and goes, 5? Really? And you think to yourself, oh dear, you know, I may not get that raise. I might not be getting that parking space I was hoping for. Maybe I should go back to the drawing board and do a better job. The problem that she's thinking of, and that you should be thinking of, is that your sample size is too small. If you're really doing this experiment to be absolutely sure that you have an effect, that this chemical X has an effect on the grass, and you want to make your customers happy, you want to make sure that you don't waste a whole bunch of the company's money bottling and bagging this chemical X for people, and then find, you know, that the 5 plants that you tested just happened to have an unusual reaction and no other plants do. So to really make sure that your experimental results are valid and that your experiment has value, you want to watch your sample size. So your sample size is something that needs to be looked at in this. So if I were to title this graph, I would say the effect of chemical X on growth rate of grass. And then I would add something. I would add an N value, and an N value is your sample size. And that tells the person reading the graph that, in fact, in your experiment, you only used 5 plants per group. 5 got 10 mils, 5 got 5 mils, 5 got 1 mil, and 5 got 0 mils. And that's all the plants you had. And you could have done better than that. Maybe it would be better if you're in a working for something that's incorporated and you have the money, go for 100 plants each. And then you're talking. Then if your data still looks like that, that's 100 plants that grew that much more than these 100 control plants. And that's much more significant than if you only had 5 of each. So you want to keep that parking space. Okay, so that's the simplest explanation of what a controlled lab experiment is, and how to design it, and how to make sure that you can do a good job on it. And I hope that's helpful.